Hello and welcome to this first video podcast in a series of 5 for the course on computer modelling of biomolecules, part of module CH4P4. On this starting screen we have a short video taken from work by De Groot and Grubmuller, in which they looked at the motion of water molecules from one side of a cell membrane to the other through a protein called aquaporin. And this is just one illustration of how molecular modelling can be used to gain insight into the structure and function of biomolecules. We'll see many more examples as we go through the course. So in this course, we'll be looking at the fundamentals of molecular modelling, so the concept of a force field. We'll be looking at the theory of molecular modelling and of course lots of applications. In particular, we'll be looking at topics including geometry optimization, normal mode analysis, Monte Carlo simulations and molecular dynamics simulations. And at the end of the course, if we have time, we'll look at some more advanced topics. There are also practical sessions with this module, so you'll have 4 timetabled workshops in the PC room in chemistry, in which we'll put some of the theory into practice. You'll actually do some hands-on molecular modelling. As always, there's some recommended reading, so we have 2 books, so Molecular Modelling by Leach and Molecular Modelling by Hinchliffe. There are copies of both of these in the library, and they're well worth a look. We will be using the primary literature quite a bit, so every week there will be a recommended paper that I expect you to read, and we'll be able to discuss this in the classroom session. And if I spot other interesting papers as we go through the course, I will post these on Blackboard. So here are some more pictures taken from work by De Groot, and these are really here just to illustrate the wide range of things you can use molecular modelling for. So in the top left here, we have a picture taken from simulations of a lipid bilayer. So we have the lipid molecules in green, and these blue molecules in the bilayer are cholesterol molecules. So by doing simulations, we can find out what the functional role of cholesterol is in biological membranes. This second picture is from simulations of a small protein called BPTI, so bovine pancreatic trypsin inhibitor, and a whole load of snapshots from the simulations have been superimposed, and these have been used to help understand the NMR spectrum of these proteins. This picture on the right looks again at the aquaporin molecule. So these yellow arrows illustrate the water dipole moment. And as the water molecule goes through the protein, there's a sort of moment at which the dipole moment has to switch round. The water molecule has to change orientation. And this helps let only water molecules go through and not other molecules. It's a key part of how it actually works. These last 2 pictures here are taken from modelling of a protein called bacteriorhodopsin, and this is a protein which is activated by light, and this part of the protein in the middle of the channel moves when light shines on it. There's a cis-trans isomerization, and it moves either out of the channel or into the channel. So molecular modelling can give you insight into a whole range of different biological systems, and in this course we'll be hopefully showing you that molecular modelling is actually much more than just making pretty pictures. So why do we do molecular modelling in the first place? Often it's actually easier, quicker and cheaper than doing experiments. Imagine if you've got a protein and you want to look at a mutant of that protein. It's a lot quicker just to do the mutation in the computer rather than to go back to your cells and have to try and produce a new protein. A nice thing about computing is that there's no significant environmental or health risks of doing computational work. Computational resources are getting cheaper and cheaper, so it's very easy now just to throw together a computational cluster and do calculations. Even on a mobile phone it's possible to do simulations. Molecular modelling is often complementary to experiments, so often we can get insight into physical processes at the molecular level from simulations which we wouldn't be able to get from experiments. The ultimate aim, of course, of molecular modelling is to actually completely remove the need for experiments. If we can model everything, then we don't actually need to do the experiments in the first place. Ultimately, we should be able to obtain all required data from calculations, but you'll be pleased to hear we're not quite there yet, so there's still room for experiments. In particular, we still need experiments to validate calculations. So if you do a calculation which predicts something, you need to be able to actually go and test that that prediction is correct. Otherwise, you don't really know whether to believe the results of simulations or not. And validation is a key part of molecular modelling, and we'll see that again as we go through the course. I've mentioned already the synergy between experiments and calculations. So experiments can suggest calculations that we need to do, and calculations can then suggest experiments which might be useful to do. So it's very much a cycle between experiments and calculations. So let's take a step back now and think a bit more about the sort of almost philosophical side of modelling. So first of all, what is actually a model? So according to the Oxford English Dictionary, a model is a simplified description of a particular system or process that can be used for a theoretical understanding or for calculations, predictions, etc. So we can straight away see how this can be applied to biomolecules, because a biomolecule modelling will be using a model to understand biological processes. In practice, a model consists of two components. So we've got a representation of the system and a set of rules governing the system. So you will find this in all models, whether they're biomolecular models or other types of models. When you choose a model, you've got to choose it depending on the purpose of the modelling. So you've got to ask yourself, why are you actually doing this modelling? What do you want to find out? And then you've got to choose a model which is appropriate for that. In particular, the model should be accurate for what you want to look at. There's no point in choosing a model which doesn't correctly reproduce something that you need it to reproduce. It needs to be efficient, so you need to be able to get results within a certain timescale, whatever that timescale might be. There's no point in coming up with a really complicated model which gets everything perfectly right, but then would require 10 years in order to do a single calculation. And finally, the model needs to be possible to validate the model. So we need to be able to predict something in a model which we can then observe experimentally, or often you've observed something experimentally and you need to check that the model reproduces this. So as I mentioned already, biomolecular modelling is all about choosing representations and rules for biomolecular systems. In molecular modelling, there's a whole hierarchy of different models that you could use, and this goes back to the point I just made about choosing a model which is appropriate for what you want to look at. So here I've got a table which I'm going to slowly build up of various systems, rules and concrete examples. So we'll start in the middle of the table. So if we say our system consists of molecules, nuclei and electrons, so the atoms made of nuclei and electrons, we could model this using the Schrödinger equation, so using quantum mechanics. So an example of this would be if you have proton transfer reactions. So in a protein such as bacteriorhodopsin, there is a proton which hops within the protein. And if you want to study this process, you're going to need to use quantum mechanics in order to model it correctly. However, if you now go to larger molecules, and you're starting to think about macromolecules, quantum mechanics is then sort of a bit too challenging because it would be too time consuming. So there are other approaches we could use instead. So typically we'd use classical mechanics, so Newton's equations of motion, and you can also use statistical mechanics to investigate these macromolecules. So an example of this might be something like the migration of oxygen, so O2, in myoglobin. So myoglobin is a protein in muscle similar to hemoglobin, which binds oxygen, but the oxygen's got to get from outside the protein to inside the protein in order to bind. So understanding this type of process, you can study using Newton's equations of motion, classical mechanics, instead of quantum mechanics. If we now go up a length scale and we're thinking about macromolecules and aggregates of macromolecules, so if we think about surfactants, if we think about micelles, vesicles, things like that, then these are getting larger and larger. And we might want to use a different type of modelling to look at these. So we could use so-called Langevin dynamics or Brownian dynamics. So that's the dynamics which describe Brownian motion. An example of this might be long timescale processes, so self-assembly processes which might take place on the sort of seconds, minutes timescale. These are out of reach of classical mechanics. Finally, as we increase the length scale even further, we can start to think about bulk materials. So we might want to think about bulk liquids, in which case we can use the rules of fluid dynamics. So if you want to work out how a liquid flows from one point to a next, but we don't actually care about what the liquid is made of or how the interactions occur between components of the liquid, then fluid dynamics does the job. So an example of this would be looking at bulk liquids and bulk gases. We can also go the other way, go to a shorter and shorter length scale. So if you go from molecules, if you go down to thinking about atomic nuclei, electrons and photons now, then we'll have to use the rules of relativistic time-dependent quantum mechanics. So these are relevant in biological systems as well, because we have a lot of proteins which do electron transfer. So things like cytochromes involved in for example, photosynthesis. If you want to study these specific reactions, then the Schrödinger equation won't work because we've now got photons coming in and doing things. So that's why we need a different set of rules. And if you go to an even shorter length scale, we could think about subatomic particles, so the quarks making up the nuclei. In this case, we'd need to use quantum electrodynamics. An example of how you might want to use this would be, for example, modelling processes going on in the Large Hadron Collider. However, here we're really into the domains of physics rather than chemistry, and we're not going to think about this any further. It is actually possible to do multi-scale modelling, and that's an area of current research, and this is when you have several levels of modelling at the same time. For example, there's a technique known as QMMM, so this stands for quantum mechanics, molecular mechanics modelling, and the idea here is that you have, for example, a region of a system which you describe using quantum mechanics. So this could be the active site of an enzyme, for example, where bonds are being made or broken, and you embed this in a region which is described by molecular mechanics. So instead of just modelling the active site by itself, then you embed the active site in the protein, but the protein as a whole is too big to model with quantum mechanics, so you need a different approach. And you can even take this system of a quantum mechanics bit and a molecular mechanics bit and embed it in an even larger bit. For example, you might have some Brownian dynamics describing how this protein diffuses in a cell, if this is important for the modelling. One of the issues with QMMM type calculations or multiscale modelling is what happens at the interface. So how does a quantum mechanical part of the system interact with the molecular mechanics part of the system? But that goes way beyond what we want to cover in this course. Okay, so we'll be focusing on so-called molecular mechanics in this course. And we'll be looking at biomolecular modelling using classical mechanics, so Newton's equations of motion, to do simulations at the atomic level, so the molecular level. This whole area is called molecular mechanics, so you'll sometimes see this abbreviated to just MM. And this is the focus of the rest of this course. And that brings me to the end of Lecture 1. So before the classroom session, you should read the associated paper for this week, which is linked on the Blackboard site.